It's a paper I got from a professor I emailed. So that's the backstory of it.
We are live, and we're going to talk to rational empiricist, or Andrew, and immutable, or Andy.
Yep. What's going on?
About a paper.
Nice.
And I'm waiting to get hammered with sound here. Oh, no, I'm not going to get hammered with sound. Good. So good. We have... what's happening?
What was that?
I love being an administrator on this channel.
Yeah.
It has its
I'm going to have to change my password.
You had me sign in already, so...
I don't want you to turn into a Gavin.
To a Gavin?
Yeah, and start putting people in the back room and muting them over and over again.
What would being a Gavin look like exactly?
It would be...
Okay.
Haven't you seen what he does to the Rage on Praise's channel?
Nope.
He mutes him every other word he says. So he can't get a word out. And it's just so ridiculous. You get tired of listening to it, and I just cut away. I mean, and Praise puts up with, and he says, Gavin, maybe you shouldn't do that. It's like, Gavin, maybe you shouldn't do that. He's like, he doesn't want to piss Gavin off because I guess Gavin pays for his channel. So kind of a problem.
Always interesting.
Yes. They are an interesting bunch, those Christians.
Interesting power couple. Well, power might be the wrong word, but you know.
Yep. Okay. So, sure.
Thanks for joining us today, Immutable, by the way.
Yeah, nice of you to show up.
Wow, you make it sound like I never show up to a stream, Michael. God. Kind of fucking rude.
Okay.
Hey, Speed, I'm late sometimes too, you know.
How many times have you showed up on time, Immutable? On time.
Well, that's not relevant, okay? We're here. I didn't miss too much, okay?
I appreciate you. Even if you're mutable.
Well, okay.
Okay. I do appreciate you. Evolution. How many phenotypes do regulatory
I'm actually not sure if this is the full paper. I don't think so. I think it's just like a sketch.
Oh, it looks like just the abstract.
Yeah.
Oh, wait a minute. Maybe this is one of those real short little. Okay. I think this is just one of those short little what do they call them? Articles or?
Yeah.
Oh, they call them dispatches. Interesting.
Go to the correspondence link.
Huh?
Go to where it says correspondence and then the DOI right under it.
Okay.
That might lead to the paper.
Let's see. Open link. Where the hell? Where's open link in new tab? Oh, I see. I'm on a PDF. I'm an idiot. Okay. Summary. That's not the paper. What? Not the same paper at all. Totally different. This is selenocysteine or something.
What?
Pseudokinase is flipping the ATP for ampylation. So I don't know what the heck. I don't know what happened.
Do we have to stick with this one then, I guess?
Yeah, I think this is the entire paper. I think it's just a real short little article, just talking about the field. They do that all the time, and
I feel like it's the best we have at this point because I don't know what else to do.
Yeah,
I feel like I saw the paper somewhere else, but I don't know what it was called. So, oh well.
I'm looking it up and trying to find out if I have it. Okay, we got it on. Is this Science? Is that on Science Direct? Okay, we got it on PubMed. And PubMed, let me see here. Full text links, affiliations. I'm trying to figure out whether or not their DOI is going to work. Well, you can go ahead and introduce it if you want.
Oh, okay. Yeah, so I guess you could say I was given this paper by someone I emailed, and what I found interesting about it was there was this gene or the details I can remember are that the gene called scute controlled the expression and development of bristles on what species of Drosophila was it? It wasn't melanogaster.
Centromia.
Yeah. It was in that family or that genus basically, and the bristles on the genitalia are controlled by the gene, and there's a specific enhancer. Like it said, there were dozens of enhancers next to it, but this one enhancer controlled the expression of the gene, and therefore the bristles on the genitalia, the abdomen of the fruit fly, but it also controlled what seemed to be bristles on specific structures on the forelimbs. I think they were called sex combs, or sexual combs or something like that. And what was interesting was, as far as I could tell, a single mutation they found interacted with the gene, or a mutation in the enhancer interacted with the gene in such a way that bristle numbers on the genitalia were decreased and bristle numbers on the forelimbs, on whatever structures they were called, were increased. And so what I found interesting was this single enhancer region of this specific gene apparently controlled two different traits, basically. So it's pleiotropic. And what I found, or what I thought was interesting about, was kind of it kind of got rid of the whole idea of loss of information, because which one's a loss of information, which one's a new feature, basically. Like how do you distinguish between the two?
Right.
That's the ultimate question that creationists never really answer properly. Because if you gain information, it's not really a gain of information, because that information always existed. But if you lose information, but if there's a decrease of information, then it is actually a decrease of information, because creationists say it is.
Right.
And
Right.
there's no consistent like standard you can hold them to say like, well, what do you mean by new? Like, if I can show you something new, like that an organism is developed but nothing is shown up there, oh well, no, that's not really new. The genetic potential for it to exist was always there. It's like, really?
Well, that's why I find this paper so interesting, is that if they're going to say that a decreased number of certain body part of bristles is loss of information, then you have to say that the other increase of bristles on the other body part was a gain of information. Like, otherwise it doesn't really make sense to say that they were both a loss of information. You know what I mean?
You would genuinely think.
Yeah, there's this confusion about how genes work. They see they see the DNA as a blueprint, and so they call all of it information. So then if they do that, they have to, for something to be a blueprint, it's instructions for some other kind of thing to build something.
Right.
And you could make a case that the exons that build a protein do that. But what do you call the regulators and the enhancers and the promoters and all of the little microRNAs and things that are running around and creating all this?
Well, I think the most common one is like a conductor of some kind.
Well, I mean, I think if you wanted to like actually try and use that argument, you would say that the like regulators and enhancers and promoters and all that are kind of like regulatory switches for the gene. So based on how they're methylated or things like that, it might turn off, it might turn on.
Yeah.
Possibilities that are permitted based upon, if a silencer is damaged, or if a silencer is being activated, or whatever.
Well, like my only issue with that is, like, even if there's a certain mutation, it's not completely on or off. It's just difference in expression.
No, typically it isn't, because typically what happens is, like CPG islands, for instance, so areas that are clustered with cytosine and guanine typically typically are methylated by methylase enzymes, which methylate on specific positions of guanine and cytosine. And typically, what that'll do is it prevents the gene from being transcribed because it messes up how transcription enzymes try to bind to that site.
Yeah, so the whole blueprint thing breaks down because what happens is the proteins are created at some concentration in each cell type, and it very much depends on the time. The generation of an organism, from single cell to adult, is going to require a temporal and spatial application of those promoters and those genes. So they're going to pop up at certain times. And there's all kinds of physical mechanisms that are used, like gradients. And there's they call it the French flag, where basically it will, because something is red, it will turn what's, to the right of it white, and to the left of it white. Lateral inhibition, it's called, that sort of thing. Or you can actually get like a 3-color pattern based on two different proteins in the organism. So anyway, it's a real complex physical spatio-temporal formula that has nothing to do with a blueprint. That isn't how we read blueprints. That isn't how you build something with a blueprint. A blueprint requires an intelligence such as us to read the information and say, oh, I need a board, I need to do this. That isn't how genes work at all.
Yeah, the issue with this statement, Otangelo, is that proteins in genetics does not operate by Boolean logic in any sense.
May I ask what that kind of logic is exactly?
Boolean logic, unless I'm misremembering, is a type of logic that only considers propositions which can be true or false.
Yeah, it's like Boolean algebra. It works with zeros and ones, so it's like binary. Something either on or off.
The thing is, though, like with genes, it's not exactly always 0 or always 1.
Yeah,
right?
Boolean logic requires the least intelligence of all. It is the most simple on-off switch type intelligence, and that's why we use it in computer chips, because it is so completely programmable, because it is so basic. It's so dumb. It's a very dumb kind of logic, on and off. So
yeah, in reality, you have, to an extent, what would be considered like partial expression, or partial truth, or things like that. Various gradations along that line between 0 and 1 for real systems.
Yeah, well, and with basically with on-off logic. Well, in the cell, it isn't Boolean logic, because it's not just on-off, because sometimes these promoters and enhancers, or these basically regulatory RNAs and proteins, do not always attach statistically, with 100% accuracy. Sometimes it's 10%. Sometimes it's hit and miss. Right. And it depends. So you end up with an analog which is not Boolean. That's anything but Boolean.
And the issue too is that what's curious about like biological systems, and no, Angelo, it's still not Boolean logic. But it's that certain times, like with enzymes, for instance, so enzymes will need cofactors and metals typically to work. But sometimes an enzyme just having the substrate it needs to carry out a reaction is not sufficient, because there's other metabolites floating around that can bind to allosteric sites and allosterically inhibit the enzyme. So what that means is that some small molecule, say carbon dioxide or whatever it is, will sometimes bind to a pocket in that molecule and cause its conformation to change, which in turn prevents the reaction from occurring, even though everything that is needed for that reaction to occur will not occur.
Right.
Because there's a change within the internal structure of that enzyme that prevents it from proceeding. So under like a Boolean algebra, you would say that everything that is needed for that to be true, there is both an effective cause and, everything is satisfied for it, but it still does not occur because there's something more to the system.
Yeah, like creationists would say that it's like an intricate system, or it's irreducibly complex or whatnot. Well, like, to me, in terms of this regulatory pathway kind of the whole deal right here, like it seems it's intricate, to be sure, but like it doesn't seem to come all at once. It seems to be built on certain layers that are built on other layers, and so on and so forth.
Tangelo, I'm really not sure you want to talk about enzymology right now, because the thing is, basic enzymes acting in succession or acting in parallel are sufficient alone to cause regulatory networks to arise. Part of that is because when you have an enzyme that takes in a substrate, that enzyme has a particular velocity in which it's able to convert the reactant to the product. That's natural. It can't go infinitely quick, but it can do good enough. So that new thing reacts with another enzyme to create some other new thing. But the issue is, because there's a limit to how quickly these reactions can take place, some of that product will end up going back into the process and binding with an enzyme in a way that causes inhibition. And this happens on several different scales with chemicals. In fact, like in glycolysis, there's certain steps where, interestingly, the product of a reaction inhibits the reaction from occurring. So I don't know where you're getting this wild claim that feedback loops don't turn into a regulatory network by an unguided process. The obvious false statement here is that you're assuming it's an unguided process, and that you are ignoring entirely the chemical aspects of what's actually at play here.
Right, and this is not logical, Tangelo. This is a consensus sequence, and what we have here is that AUG, that is fixed. That has to be there. But now, like, if we look, can you see my mouse? If we look one over here, okay, that could be it's most likely a G. It will fit better if it's a G. And if we go over to this side, it's going to fit really much better if it's a C. But it could be a G, an A, or a U. So, I mean, these things can all have different values, and that thing is still going to hit that sequence. So when you have a promoter for a gene expression, there's going to be this sequence out here that can mutate. And when it mutates, it's going to hit that gene a certain percentage less or more than it did before. So it's going to change. It's going to more closely match this consensus sequence.
Oh, Angelo. I really want you to come into the room right now and just tell me what I'm wrong about in terms of enzymology.
Not read the relevant. Let it. Oh no, he's going to link Cat's board. He's going to link Cat's board.
Oh dude.
Okay. Here's a fun little thing for you. I really don't care about when you choose to cite your own work as if though it's an authority.
Yeah.
I'm actually concerned, like, when you choose to try and read scientific articles, and the arguments that you make are so asinine and the reason I can tell they're asinine is because you are citing every time, often, if not almost every time, from the abstract of a paper.
Right.
Which means literally nothing. Because the purpose of an abstract in a scientific paper is to explain what the problem is, what method the researchers were using to address said problem, and how that problem was then solved.
Otangelo, you're building you have this stuck in your brain that these networks have got to have intelligence to create them, and they do not. If an intelligence created this thing, it would operate a whole lot cleaner than what I just showed you. It wouldn't work on a consensus sequence.
And here's the thing, not even going into the science of it all, but by Otangelo's very logic, right? It would be quite literally impossible for this God figure, or whatever it is he wants to call it, to have formed itself, especially being that it's a being of infinite complexity. Right? Because the claim on behalf of people making this argument is that God is something that is eternal and has existed forever. And there's nothing responsible for its creation. Okay, that seems to be, to me at least, inconsistent.
Okay, so on this paper, let's see. Did you want to go over a little more of it, Andrew, and maybe report?
Sure. Wait, was Otangelo here the whole time? Did he catch the question I posed?
I don't know. I think he
we
came
ask
in
him?
a few minutes late.
Should we ask him which counts as a loss of information?
Yeah, go ahead.
And I'd want him to come in because I'm curious where he thinks the hell I'm wrong.
Yeah. I mean,
that way Doom lines.
Otangelo wants to come in. I'm game.
Well,
if not, we'll see if he says anything in the comments.
Okay, I can open this up by putting the link in the chat, and
Okay.
I do so only with your approval and with your okay.
Okay.
Similarities in the processing of information by biological systems and human-designed devices are broadly recognized by many researchers. Yes, and all of them, almost all of them, will say it is the result of complex biochemical pathways that are interacting in a very complicated way. There is no intelligence that is interfering with the process at all.
Oh no, are we going to set Otangelo and Demeter after each other again?
Might as well.
Okay.
Hello,
That's control. Hi, Otangelo.
Hi, guys.
So how is that logic?
Okay, I will put here the link at my library, and you
why
can
don't you tell us? Why don't you tell us how you insist, or how you get from it's a logical network, or it's a network of influences, to it has to be intelligent designed? How do you make that implication? What is your argument to make that?
Okay, my argument is a syllogism. I can also show that if you want. I can put it in a page here.
I know what a syllogism is. Oh, you had that syllogism.
Yeah, I have it here at my.
I will tear it apart for you.
My library. I can link it here. Just a moment, please.
After that,
let me check here. I have to go to Chrome. Okay, here it is. Let me just put it bigger here.
Also, Otangelo, with your consent, I would like to ask a question based on the paper that was shown. Whenever we're done with this subject, if that's okay.
Boy. Another post from your website. Here we go.
No, I can put directly the link to the Nature article or science paper, which confirms what I am saying. I mean, I am not making things up. What I am saying is based on peer-reviewed science papers. So since you guys asked me what since Speed asked me, so I'm putting here the syllogism, and then I can back it up with that paper in Science.
Can you make it bigger? Let's see here. Well, can you link it in the chat, and then I can follow along?
Yeah, just a moment.
I'll put it not in private chat, but put it in. You have a wrench, right? Yeah, you do. Put it in regular chat.
Okay, just a moment.
If you can do that. Either way, I can work with it.
Okay, it is here in the private, and I'll put it in the regular chat as well, okay?
Okay. Let's see here. Let me see if I can link it. Oh, wait a minute. No, that's not a link.
Oh, sorry. Yeah, just a moment.
Okay.
Not the link.
Oh, yeah, I put the link now. Sorry. I take the wrong one. Here it goes. Okay.
There we go.
We got it in private. Okay.
Yeah, I'll put it here for the chat as well.
So I can pull that up over here and take a look. The problem with syllogisms is, the people that write them really think that they're powerful.
No, I wrote it based on my studies on the gene regulatory network.
Yeah, the logic makes
This is the
sense, but
result.
the premises often are not. It's what do you call it? It's valid, but it's not sound normally.
Well, the premise is based on what I have read in science papers. So
Okay, let's see. Syllogistic arguments. So, I got it, the gaps. I'm trying to find that. Why am I not finding it? I'm looking at your link. Okay, you got logic, gates, you got...
just a moment. I'll put it up here. Okay, here it goes.
God, I'm trying to find it.
Okay, it starts from 1 here. Complex multicellular life forms depend on gene regulatory networks, which are a collection of molecular regulators that interact with each other and with other substances in the cell to orchestrate the expression of DNA. GRNN operate based on logic gates, and their networks process chemical input signals similar to computers. These encoded instructions are based on Boolean logic. Logic depends on reason. Reason depends on intelligence. Only an intelligent mind can think rationally and implement a system based on conceptual laws of logic. Therefore, the best and most reasonable explanation for the existence of complex gene regulatory networks based on Boolean logic, essential for the make of complex multicellular organisms, is the creative action of a powerful, transcendent, intelligent creator. So that's the first syllogism.
Okay, I wish I could read that because I can't find it in this document.
Yeah, it's under developmental gene regulatory networks, DGRN.
So I did link to the science paper. If you want the link to the argument, it's here. I'm just linking to it now. Okay. So I've put here, the first link is to the science paper, which backs up my claim that gene regulatory networks work on Boolean logic. And the second is the inference, which I made the syllogism arguments.
Okay, so it's this last one. It starts with regulating, governing, and controlling. No, complex.
Yes, that is a video which I made, and the last two syllogisms are about the gene regulatory network.
Okay, so you're listing a bunch of things, elaborating strategies, guiding. Okay, by reading the right one, it starts with regulating, or are you starting?
You can use that one. Yes, that has to do with the gene regulatory network. You can use it. You can read it.
Sense as, let's see, our tasks of gene regulatory, which are ultra sophisticated communication networks analogous, not really analogous to electric circuits, but in the same sense as copper, plastic, basic electronic parts do not turn into printed circuit. Okay, so you just got, you've got assertions in here like, okay, basic electronic parts have nothing to do with it because there are no electronic. Just because it's analogous doesn't mean there's electronic parts in the cell.
No, I'm not saying that. What I am saying is that the structure of the gene regulatory network is like an electronic. Yes, I'm using an analogy to illustrate that we have analogies here. I mean, we are talking here about networks which have to be interconnected together in the right way and which use Boolean logic, and you have to explain how that emerged.
Yeah, but it doesn't use Boolean logic.
It does. It does. Okay, so you can check. Okay, Mike, you can check here the first link, and there I'm backing up what I am saying. If you want to open it up, I can remove that and put.
Yes, there are some regulatory networks that you can draw out to make them look like it's Boolean logic, but it's always with a certain percentage of that gene, and that gene diffuses across the cell or across the organism. There's diffusion, there's energetics, there's we lose Otangelo?
I think the meter joined, or maybe Leo.
Oh, did you take Otangelo's syllogism down? Or his paper?
No, it's still up there, I think.
Take it down.
Yeah, there it is.
Yeah, you can draw regulatory networks that way, but that isn't how they actually work in the cell.
Well, here you have it here.
But it doesn't always work that way. No.
Well,
these are actually these are actually Boolean electronic gates. Okay, nothing in the cell works that clearly exactly like that. There's always diffusion. There's thermodynamics. There's it's a stochastic project. It doesn't always fire exactly like this. For the most part, like sometimes you'll end up with so much of an enzyme or a protein that it will almost always, create this result. But that isn't always the case. And in fact, in development,
Mike, I think you are a little bit confused. We are not talking about proteins. We are talking about the gene regulatory network, which actually regulates the expression of genes, which is an entirely different topic and field. So what are we talking about here right now?
Okay, how are they regulated? What regulates these genes?
The gene regulatory network, which uses Boolean logic.
Okay, but what does the gene regulatory network do that with? What does it use?
How do genes regulate each other?
Right.
Okay, the gene regulatory networks receives inputs by signaling networks from the environment, or it adapts its output depending of the requirement of the organism of food, or also in regards of the development of the organism. So these are the three main inputs which upon which the gene regulatory network will orchestrate gene expression.
But where
Good. You said that it is involved with regulatory networks. Where do those regulatory networks come from? Because to my knowledge, those are what we call proteins.
Right.
Proteins serve a regulatory function. Proteins serve a regulatory function because of the fact that certain metabolites and certain substrates for those proteins either cause them to do what they're supposed to do or do not allow them to do what they're supposed to do. So again, it does come back to proteins, because what we are describing in terms of some genes that are expressed, some genes are directly related with the activity of specific proteins and the ability of metabolites to allow those genes to be expressed.
Well, the thing is that, of course, you need proteins which will turn on and off the genes.
Yeah.
But and yes, of course, they are also made by genes. So we have, in a certain sense, here a circuit, or how can I say, a circle where you need I forgot the name. You need the proteins. I forgot the name of these proteins to express the genes, but you need the gene expression to make also these proteins. So what came first?
Do you mean transcription factors?
Yes, transcription factors, exactly. So the genetic information makes transcription factors, but you need transcription factors to orchestrate gene expression. So of course you have to ask now, what emerged first?
Well, the original pathway, the transcription factors that got kickstarted basically.
Hold on.
We have an analogy by Dr. Dan, by the way.
The issue here is we are not talking about the origin of that process. We are talking about a genetic system that is influenced and directed by a system of protein regulation that is very non-Boolean. And genetics itself is non-Boolean, because in the same way that proteins operate, there are certain transcription factors needed in order to express certain genes that, even when they are present, are oftentimes and sometimes not able to be expressed. Because even though the promoter and enhancer regions for those proteins is available, it is not always possible for certain factors to bind to those regions and to express and transcribe that region. So again, this whole thing about it being Boolean, of just being it is expressing or it's not expressing, is not correct. Because there are other pressures which are acting on certain genes that dictate whether or not they will be expressed, or whether or not they will, or whether they will or will not be expressed.
Well, we are directly contradicting what I'm reading in that science paper of Nature magazine. So I don't think that you know more than they do.
What paper? What paper?
Okay, I will link the paper so you can look it up. You can
Thank you.
Open it up. Okay, just a moment, please.
We can't just talk about papers like we just made them up. They're not like Santa Claus. We have to show them. I'm
Okay.
sorry. I'm a bit of a stickler for that.
Immutable, just so I may clarify something, so you were talking about promoters, enhancers. What was the other element you were talking about? Silencers, essentially, that prevent
Yeah,
the
silencers.
binding sites.
Yeah, silencers are elements within, that are pre-genic elements that prevent protein from being
Okay,
expressed.
here's the paper, Mike. If you want, you can put it on the screen. I'll put it here also on the side.
Let's see if I can find it here. Weissman.
And thank you for joining us, Demeter.
Oh, so okay. So wait a minute. What?
I'm just listening.
No
If you want to bite in here, feel free, because this is going to be an interesting.
I don't want to be a nasty guy.
Networks.
It's okay.
Share screen.
Yes. Hi, Dimitar.
Hi.
Should we show you the paper too?
Oh my goodness. Network motifs. Okay. Share. Okay, so I got it.
There we go.
Maybe I can make it a little bit bigger. Let me see if I can make it bigger without. Okay, so can everybody read it?
Yep.
Is it good? Okay. Control the expression of genes. As transcription networks, well-studied microorganisms appear to be made up of a small set of recurring regulation patterns called network motifs. Diverse organisms, right? So they're reused all throughout evolution. Okay, what part of this paper seems to be the part that you want to talk about?
Just a moment. I'll give you the page. Okay.
Ooh, it's a big one.
Okay, it's on page 3.
Page 3. So I'm on page 1, 2, 3. Par? Starts at that?
Yeah, you see the picture, forward loops.
Yep. It's like a family and effort. Okay, feed forward loop. FFL, it appears in hundreds of gene systems. Let's see. So we have, is this the illustration down here? For, let's see, incoherent type 4. Coherent? What are they talking about? Feed forward loop. Coherent feed forward loops. Okay, so there are feed forward loops. We know that. To understand the
Okay.
fun.
Okay. I'm sorry. Let me give you the right paper, which talks about Boolean logic. It's the same thing which in this paper, but here you have it more explicit. Can you open the last link, which I'm linking to right now, please?
Okay. That is. Let me see if I can. Let's see if I can download that as a PDF. I can. Cool. Okay. Synthesizing. Okay, logic gates. I mean, okay. Frustrated. Let me see. NIMS 2437. Okay. And we got to make it bigger. Open and crash my Mac. Got a lot of things open today. Okay.
Damn Windows.
Huh?
Close some damn windows then.
Oh, I can't. I have so many things going on. I can't. I close my windows when my Mac crashes. Okay. Okay. Distracted by Dan. Okay, what do we got here? Synthesizing biomolecular-based Boolean logic gates. Okay, so Recent avenues, synthetic biology. So we're talking about synthetic biology here, and they're using the idea of basically using gene regulatory networks to create things. I mean,
if you go to the one after the abstract, at the second block of writings, they write here, digital circuits make extensive use of logic elements which are interconnected to create logic gates, capable of executing Boolean logic functions, including not, or, and all their possible combinations. And then you go a little bit, scroll down, and then you will see these combinations. That's exactly what we have also in the gene regulatory network.
Why am I not? Am I in the right paper? I'm not finding what you're reading.
Scroll down a little bit.
Okay. So I think we're out of the abstract.
Okay, here we are.
And I have the paper that's linked. I put it up on Sci-Hub. So
Mathematically processed into a digital signal as a code representation and physical cues. For instance. Yep. So they're making an analogy to a Boolean, to a digital computer, and they're talking about how they could do this with enzymes. And this is synthetic biology. And these gates, the sensors read out inputs, and then the computational core assigns them a value of either 0 or 1, depending on the threshold set in place. Okay. Depending on the threshold, now that's analog. Thresholds are analog. Combination of these values meets the system. So what are they trying to do?
They are basically copying the gene regulatory network and its functions and making a synthetic computer through that.
So they're actually, if we look up the abstract here, main components of a computing device. Okay, these parts indeed, with the advent of more sophisticated tools, we have these devices function in the context of either test tubes or living cells and yield highly specific outputs of a given set of inputs. Yes. Okay, so they learned how to do with very small chemicals and enzymes what we've been doing with digital circuits.
They are basically basically applying biomimetics. They are taking what happens in the gene regulatory network and try to copy it.
Okay, so we've got an analogy here, but I don't understand how you get to it. Has to be intelligently designed.
Well,
obviously,
then
these guys are
the leap to intelligent design. First, you have to establish that the gene regulatory network indeed uses Boolean logic in order to make decisions when and where to express a gene. That is a scientific fact.
No,
now,
that isn't a fact. It doesn't use Boolean logic. It's analog. It can be mapped out as Boolean logic, and sometimes there is so much of a concentration that it is almost like a 1 or a 0, or so much or so little that it will actually flip all the way to one side. And sometimes there are feedback loops where something will create a protein, and that protein will create more of itself, and so it flips all the way to a 1. And in that sense, it's kind of like a Boolean, like a computer flip-flop, or it can flip back to a 0 by inhibiting itself. So you have analogies here. How do you get to this? It has to be intelligently designed.
Okay. I read at my page, Boolean spatial output. Animal bodies display a fundamentally Boolean character at every level of organization.
No.
So,
no.
Well,
no.
I am literally reading here from a book, Genomic Control Process Development and Evolution. For these reasons, except where they are shown to directly cause spatially animal bodies, display a fundamentally Boolean character at every level of organization. So, and I have another science paper here which says that. So you would have to argue actually with my source, not with me. Okay.
Your source isn't saying your source doesn't say it was intelligently designed.
Of course
your
not,
sources.
Mike. Of course not, Mike.
Okay, what was that
What
book?
I am doing here is following. First of all, I am establishing the evidence. What I am claiming is that gene regulatory networks use indeed Boolean logic for orchestrating genes and expression of genes. That is the premise. And of course, now you have to ask, what is the best explanation of the origin of such regulation based on logic gates? And from there, I go and make my syllogism, which is my inference. Of course, you can disagree and say, well, evolution explains it better. But I cannot understand how you can come to that conclusion, because you need actually foresight in order to make such a gene regulatory network, which is integrated, complex. It has to work together with genes. So you have to have all this all at once. You cannot have it in a stepwise manner. If you don't have the gene regulatory network, how do you express genes? How do you orchestrate the whole expression? How do you actually build an organism without it? You can't.
Uh-oh, Leo's here. Okay, now,
So,
Angela.
can I enter a little bit, or just you want to say something?
Oh, no, go ahead, Demeter.
So, Otangelo, this is your style of arguments. In general, you're looking something that resemble whatever intelligence people do, like computers, cars, calls, systems. And of course, because just intelligence do that, therefore it's logical, right, to be intelligently designed. Am I right here?
Yes, I am making a positive argument for intelligent design,
Okay,
which is
so
not
that's
based
your
on
logic.
ignorance.
So this is your logic. It's very resemble what we humans do, right?
Yes, correct.
So, and it's logical to be intelligently designed, right?
Well, it is a logical inference, yes.
Yeah, that's logical. Okay, so going outside, outdoor, and you see every day the sun, how it's rise from east and sunset on the west. And now it's very logical, based on this observation, to assume that the sun actually orbits the earth, right? It's super logical. It's absolutely logical. Now, the science doesn't work only in logic, Atangel. Doesn't look the logic, yes, but that doesn't mean it's a final conclusion, because there are lots of things that are absolutely logical and happen to be not correct. As I said, if you see the sun how it's rising from east and sunset going in the west, every day is doing this. The logical explanation is that the sun circled the earth. It's so obvious. So see what I'm going? Yes, it's a logical to assume, but it's not enough to finish your thought. Okay, go ahead, Michael.
Okay, so I wanted to ask you a question. So you have these sources that are saying it's a Boolean circuit, it's electronic circuit, it's a digital circuit. You have all these papers. Why is it that none of them concluded that it was intelligent design, but you have? What is different about you and them?
Well,
and
we have actually talked a little bit with the other day with the other guy about the history of science. Okay, and I can quote here why I think that is the case. Okay, like for example, Nobel laureate Christian de Duve, he says scientific inquiry rests on the notion that all manifestations in the universe are explainable in natural terms without supernatural intervention. So this is also an a priori assumption, and then you start already the scientific inquiry by assuming that you cannot bring intelligent design or a supernatural cause into the explanatory framework. And then you have to start with material causes and end with material causes, and only search material causes to explain a phenomena which is observed in the natural world.
Well, you're
explicitly wrong. Science
True.
excludes anything that doesn't provide explanations, that cannot be tested for, and that doesn't make novel, testable predictions. Anything. It doesn't matter what it is. If it can't do those, it doesn't matter to science. It just so happens intelligent design falls into that category. You are bringing to the table an unfalsifiable assertion. Because it is unfalsifiable, by definition, it can't be science. Science is not making an a priori judgment. They're saying, look, if we can't work with this, we won't try to. And that's what's happening.
And just to add on to that, like if you're gonna say it's a supernatural explanation like intelligent design, that's on the other hand assuming that it's everything except natural. How do you know that it's something natural that you haven't thought of yet? That's an argument from ignorance.
Also, I want to mention, in addition to that, yes, we are assuming that the natural is the explanation. That's our assumption. However, we are constantly testing this assumption. So, and is it this assumption is proven by experiment, hypothesis experiment, or logic based on a law, based on known interactions? Therefore, there is no need to have anything not logical and this assumption not to work, not to be accepted. Meaning, as long as we are getting, without need of intelligent designer, everything is fine. We don't need intelligent designer to take it in account.
Also, I did want to jump in really quickly. This is why I hopped into the panel, into the stream, to talk about this Boolean logic in GRNs. So, Otangelo, you tell me if this is a reasonable summation of your argument, that you are arguing that GRNs, or genetic regulatory networks, operate on a Boolean logic model, and there had to have been some intelligence that wrote that logic into the GRNs. Is that accurate?
You're muted, Otangelo. Oh, Otangelo's still muted.
Oh dear.
Maybe Otangelo had to step away.
Perhaps.
Anyway.
I'll wait till he comes back then.
Okay, sorry guys, I've
Oh.
been on toilet.
Did you hear what I said?
No, I was.
Okay.
on the
So
restroom,
I'm gonna try to steelman the argument that you're making, and I want you to tell me if it's accurate. Your argument is that in GRNs, or genetic regulatory networks, that there's essentially Boolean logic written into how genetic regulatory networks operate, and because of that, some sort of intelligence had to write that logic into GRNs and how they work. Is that accurate?
That's my inference, correct.
Okay, so that is not correct. And a paper here from the 25th of September 2018 from the Center for Advanced Computational Solutions at Lincoln University in New Zealand, written by Leshi Chen, Dan Kulasiri, and Sandhya Samarasinghe. And I just wanted to quote this little bit from this article. Boolean modeling is the simplest model for GRNs without the need to consider any effects at the intermediate levels. This modeling was initially introduced by Kaufman in 1969, following the discovery of the first gene regulatory mechanisms in bacteria. Since then, Boolean networks have been intensively used for modeling gene regulation. A Boolean model is constructed using Boolean variables in either of two binary states that represent gene activation or inhibition, respectively. Each variable represents a gene included in the GRNs with its next state affected by a Boolean function. A Boolean function, denoted by F, is a logic rule that gives a Boolean value of 0 or 1 as an output based on the logic calculation of the Boolean input as defined in equation 1. So The Boolean logic is not written into GRNs. It's a way of modeling how they work. That's it.
Yeah.
Oh, what was the name
And
of
I
that
can
paper,
cite this
Leo?
paper, which was published in Frontiers in Physiology.
What is the name of that paper?
I'll put it in the private. I'll just link it in the private chat.
Okay. You guys are killing me with this private chat. You realize StreamYard throws away private chat as soon as you leave.
It
Yeah,
exists
it
for a reason, Michael. I'm sorry to say
What?
it.
What?
It exists for a reason.
Yeah, but it's private and
Oh my God! Now
it sucks. And people that use private chat suck.
Oh, by the way, the name of the paper was A Novel Data-Driven Boolean Model for Genetic Regulatory Networks.
Okay.
So what they're doing is they're utilizing a system that we have developed, because Boolean, named after who invented it literally, is simply a model. It's a way that we can model the operation of GRNs. It doesn't tell them what to do. It just describes what they do.
Yeah. Okay.
Like pretty much everything that humans develop.
Well, Leo is making the basic mistake again, which he is doing also at other places. Of course, we are discovering that the gene regulatory network uses Boolean logic, but
We're not discovering that.
Well, Leo, okay, I will recommend you following. When I am talking and when others talk, please do not overtalk. Let people finish to talk.
Well, then please
Because
do not
otherwise,
misrepresent what I'm saying.
otherwise it becomes very annoying, and I am not very keen
Yes.
to
is annoying
have,
when
to
people
have
misrepresent what I'm
interactions.
saying.
Go on.
To have,
Go
to
on.
have interactions when I am constantly being interrupted, especially by you, Leo. So I ask you a little bit to wait until I finish, and I will grant you the same respect. Okay? So in implementing function of logic gates, that is always something that is made by intelligence. If you have evidence that unguided, non-intelligent mechanisms can implement computing by input and output using Boolean logic, be my guest and give me the evidence. I will be very happy to see how that works, how natural unguided events can establish computing.
so again with the same fallacy.
So, number one, you're correct. Logic is developed by an intelligence. That intelligence is humans. Humans can then take the very thing that they developed and apply it in a modelistic form to reality in numerous aspects of it to describe how it works. That's literally what science is and what science does.
As said, we have discovered that the gene regulatory network works based on Boolean logic, and you have to explain how that was. You have to explain how that was implemented. And I do not know of any other mechanism than intelligence which can use that kind of logic and computing of input and output using algorithms and logic gates. How that can be established by non-intelligent mechanisms. I have no example of this. I don't know how that could be an emergent property of matter.
Because that isn't what they're doing. That's how we describe what
that's wrong.
They're doing. Well, I just cited a paper from one of the most well-respected physiological publications on the planet that disagrees with you.
Oh no, that's a fact that the gene regulatory network takes decision based on input, and based on that input, it uses Boolean logic to generate an output, which then regulates how genes are expressed. That is a scientific fact.
That is not what it does. That's how we describe what it does.
Okay, whatever. Well,
so let me ask you a question. Let me ask you a quick question. Do black holes eat things?
We are not talking about.
It doesn't matter. It's called an analogy. You should be well versed in this because you use them frequently. Do black holes eat things?
I don't know. I'm not versed.
Well, I mean, is
a
black hole a human? Does it have a mouth? Does it digest? Obviously, the answer is no. Black holes don't eat things. That's just an analogical description that we give to them, just like here.
Well, if it is described, it's because that's what happens in the gene regulatory networks. It makes decision of 0, of 1.
There's no decisions made in GRN.
It combines information from various sources to then generate an output. And that is basically what it does, which you are denying. So you are simply not understanding what it is. Leo, why do you not keep to astronomy and astrophysics? Because there it seems that you have better understanding. Because in biology, because in biology, I mean,
that's an ad hom there, Otangelo.
I mean, if I didn't have an understanding of this, why is technical literature agreeing with my takes on it?
They don't.
And this is only there is. I just cited it. And this was
they
only
describe.
The
well, they describe, of course, what they have discovered there, and what they have discovered is
that literally is not what the paper that I'm looking at says. Literally is not what. Like the title of it is a novel data-driven Boolean model for genetic regulatory networks. The fucking title refutes what you're saying. This is a way to model the operation of GRNs. That's it. End of story. Goodbye. The end.
Hey guys, let's see, some housekeeping. Are we going to be doing that after show thing tonight, Leo?
I'm assuming so. I wasn't the one that set it up.
Okay. Are we doing that?
I'm assuming so.
Sure, I guess so. I mean,
what time are we doing that?
In less than an hour.
As long as it's there, I will be there. As long as it's up, I'll be there.
Okay, I'm going to step away for about 45 minutes.
Go ahead.
And let you guys have at it here.
Sure.
But I'm going to hop out, okay? And you are in charge. So I'm going to stop my screen.
Don't forget to take the
what?
Underneath the what's covering me a little bit.
Oh, we got to get you on top. We'll put you on top. How about that? You feel better now?
Okay.
Okay. Yep. I'm going to well, if I remove myself, what will happen? I don't know.
As long as somebody else in here has administrative powers.
Yeah, I better leave myself on.
So, I guess, Otangelo, did you, may I still ask my question from earlier? That's okay.
What was your question, please?
So the paper we were looking at earlier, with the enhancer mutations, would you consider, I guess, I don't know if you were you here for that segment? About the Drosophila?
I'm sorry, I'm a little bit confused now with your question.
Essentially, he's asking what type of changes in genetics do you think would constitute new information?
Yeah, essentially. Sorry, I'm trying to phrase it correctly.
So you are asking if mutations are new information?
Well, like what exactly would constitute new information in terms of mutations? Like what example would you say, okay, this is an example of new information being added to the genome?
Well, I mean, in order to have a new trait, you need new information, correct?
Right.
Yeah. You both answered. Is it correct or false?
Yeah, it's correct.
Okay, so let's suppose that you have an organism which doesn't have limbs, and you want to grow a new limb. What do you require?
Well, it can be change in expression of a particular gene. It can be a duplication of a gene. It can be a lot of different things.
So you think that would be sufficient to grow a new limb?
Yeah.
Well, because the exact same genes are used in both the precursors to limbs, aka fins, and tetrapod limbs, in terms of regulating expression. Like darn it, I don't remember what the specific genes were. Do you have any ideas, Immutable?
No, I don't think so.
Okay, so let's suppose that... Yeah,
sorry. Oh, go ahead.
Okay, so let's suppose that you want to grow a new limb. I will mention here 7 systems, physiological systems. Please tell me which of those is not required in that new member, okay? First, the muscular system. Second, the skeletal system. Third, the nervous system. Fourth, the endocrine system. Fifth, the circulatory system. Sixth, the integumentary system. And seventh, the lymphatic system. Do you think all these systems are required to grow a new limb, or can you grow a new limb without one of those?
No, they aren't all required.
So which one is not required?
Well, you can choose any of them really, but for sake of example, the circulatory system.
Okay, so let's remove the circulatory system. Do you need all the other 6?
Probably not.
So which one do you not require as well?
Probably not any of them really.
Yeah, so you can grow a limb with which one of those 7?
Why do you think that those limbs can't be produced by the result of gene activation and genes together in development?
So let's suppose that a gene adds new information to make a muscular system of a new limb.
Okay.
If you don't have the other 6 parts, can that new limb grow only like being muscles?
Sure, it can.
Yeah.
Yeah.
You don't need a skeletal support in order for these muscles to work.
If you don't have a skeletal system, what's constraining the growth of those muscles? Exactly. Right.
So where are
Aren't there animals that have
these
muscles without a skeletal structure there?
Is there a requirement of the muscle to be able to grow while the skeletal system is in place?
So where do you attach these muscles if you don't have the skeletal and the skeletal system?
Well, like there are certain, like many different species of invertebrates that have muscles but don't have or even cartilaginous skeletons. So I don't really understand what you're asking.
Well, what I am saying is that in order to have a new limb, you need all these systems. How do you supply blood to that muscular system if you don't have as well the growing of the circular system to supply that muscles with blood?
Are you assuming that the limb just pops out of somebody?
Hold on. Otangelo, what is responsible for the transport of blood in organisms?
The circular system.
Yes, but more importantly, what does the circulatory system use? Because there are organisms without circulatory systems that still manage to cause blood to flow around in their system. And the answer, because I suspect you don't know what I'm referring to, are proteins, which are called globin proteins.
Yeah.
Yes, you need blood with all the members of the different cells in the blood, correct?
Not necessarily.
Right.
Globin proteins are more so associated in the activity of iron-bearing of proteins that usually constitute iron in order to usually transport oxygen from point A to point B in a system. So it's less to do with blood and more about the activity of the protein. But different variations of that protein are responsible for different types of what you generally consider blood. So
And also sorry.
But yeah, so it's not dependent on the circulatory system existing to have blood or blood-like substances.
Yes, of course we have insects which do not have a circulatory system. They have a different diffusion of blood, but we are talking about the growing of limbs of a mammal, for example, to grow an arm or something like that.
Right. So
why do you mention circulatory system?
Sorry, you guys talked over each other. I didn't understand the question.
Sorry. Go ahead, Immutable.
Right. But so, I mean, why was your initial example to say that the activity of the circulatory system or of a different system is reliant on another system, like the integumentary system and things like that?
Right. That's what I said. Well,
Can I share my screen really quickly? Something from Dr. Dan.
Yeah.
Sure.
You're probably going to have to explain it, Immutable, because I'm not going to understand what it's saying, but there it is.
Which
I don't
one? In the private chat or where?
No, I'm sharing it.
Oh, okay. But for some reason I can't. Hold on. Why is it not letting me see it? Hold on.
There we go.
What is it? I can't. Oh, there it is. I couldn't find the little cursor thing. Check fetus. Okay, I'm not exactly sure what that is either. I'm not. I'm not entirely certain what that represents either. But I mean, relating back to the original point of Tangela's original claim seemed to be that there are certain systems that can only function when certain other systems are present, and that doesn't appear to be correct.
Well, I can give you an example, and just give me a minute, and I will show it here on the screen. Okay?
Sure.
Okay.
Sure.
I know
You can talk while I'm searching here.
I know exactly where Tangelo is going, but I'm patiently just listening. Let's go to the end.
I mean, there's a lot of favorite tropes examples that creationists like to use. So let's see what he pulls up.
So let's Tangelo explain first. Usually, what Tangelo is presenting, or in most of the creationists, they're presenting completely wrong model of evolution. So once you present the wrong model of evolution, they try to overrun the wrong model. But Tangelo, go ahead. Let's see first.
Well, Otangelo was talking about a circulatory system,
Okay.
and here's a slime mold
here.
that does it without anything other than the circulatory system itself, almost
Okay.
on my screen.
Okay.
Yeah.
So it makes
Dr.
no
Dan,
sense what he was talking about.
I'll drop the link in the chat for him.
Is only speed being able to share my screen?
No, I don't see it. I don't see where you're. Oh, there it is. I'm sorry.
Okay. Okay. Here you have an example of an irreducible system. That is, you have here the muscle surrounding the human eye, and the superior oblique is a muscle which produces eye movements and helps internally rotating the eye. The trochlea is a pulley-like structure close to the eye, and the muscle tendon hooks around it. You can see that here. Can you see my cursor here showing it?
Yeah, I think so.
Okay. Then the tendon approaches the eyeball from the front. The tendon, that is that part here. That is the tendon where the muscle here is attached. So you have the pulley, you have the muscle, you have the tendon, and you have the trochlear nerve innervates the superior oblique muscle of the eye. That can be seen here. Here, the trochlear nerve. Okay? And in the end, you have the vascular blood supply from the lateral muscular branch of the ophthalmic artery. So the blood supply is supplying blood to the muscle. So you have here 1, 2, 3, 4, 5 integrated systems which work in a joint venture together, where if you remove one of these parts, the system doesn't work. So if you remove the muscle, you cannot move the eye. But in order to have the muscle functioning, you need the tendon, you need the trochlea, which is holding the tendon here on the skeleton on the upside here, and you need the blood supply for the muscle to work and the nerve system. So if
Somebody
one of those is
the
missing,
highlighted comment.
So
one
if
second. I don't see where it's highlighted, but okay.
so, you guys can try to explain how each of those individually would have evolved if they do not have any function individually whatsoever, only as an integrated system.
That's not true. They do have functions
That's not-
individually.
I'm going to shut up and let Dr. Dan take that.
Well, can we go back to
Dr.
limbs for a second, though? Because what Atangelo spent like a solid 5 minutes describing about limb development was just not how limbs develop at all.
Ed.
The way he described it was that, well, you need a separate thing, right, to say put bone here, put blood vessels here, put muscles there. And that's just not how it works. It's limb development starts really early in development with really simple signals that basically say grow outwards at this site. And the pattern for growing outwards, what you're doing is you're basically taking the longitudinal axis of the body with your germ layers that form into bone and muscle and skin and everything. And what you're doing is basically just turning that arrangement perpendicular and growing it away from the body. It's just one signal that says, turn here and keep growing in that direction. You don't have to individually say, put bone and muscle and this and that. You don't have to individually say, put all this stuff here. You're just taking what already exists and just turning it and having it grow outwards. It's not that you need, he said, which of these 6 systems don't you need? That's not how it works. And you can look at like the gene regulatory networks that deal with this. And we've actually figured out like the specific genes that are responsible for it. And that link I put in the chat with the picture with the mouse and the snake gene expression patterns, that just shows 3 genes and how the 2 that suppress limb development, in mice, they're expressed in between the limbs, and in snakes, they're expressed through the whole body. So you suppress limb development throughout the entire body.
Sense.
Like it's actually very similar to that paper that Atangelo showed before with gene regulatory networks, and how the authors said that it looks like you have these complex networks, but you have the modules that are the subcomponents, and it's just variations on those modules being copied and turned around, and it's a few very simple, smaller pieces that are then interacting in different, more complex ways as you build more complex systems. Morphologically, that's basically how development works. It's not that you need to have a set of genes to build a left forelimb and a right forelimb and a left hind limb and a right hind limb. It's you have genes that say, put a limb here. And then those genes are expressed for different amounts of time during development. And that's how you get your different limbs in different parts of the embryo. It's not nearly as complicated as Atangelo is describing.
So, Dr. Dan, if I may clarify on something.
Please.
Awesome.
If, for example, or I'm assuming that you would accept that at least a great fraction or a certain percentage of the kind of mutations out there would be regulatory because they tend to be less pleiotropic, something along those lines, that they would be essential to morphological evolution because they're like you said, they're compartmentalized, basically. Would that be a safe thing to say?
I don't want to say in general, because I'm not a developmental biologist, so I don't know enough to say in general, but I can see the logic of what you're saying, that if you limit a mutation to a regulatory mutation that has a specific effect and in a specific time during development and a specific place in the embryo during development, then I can see the logic of that being less likely to have other consequences, right? I can see the logic of what you're saying. I don't want to say that I know for sure that is the case, because developmental biology is not my specialty, but the logic of what you're saying makes sense.
Okay, I just want to make sure and see your position in terms of coding mutations versus regulatory mutations, if you thought one was more significant over the other, but I'm assuming that both are important
for big. Like if you're talking like body plan changes, then developmental regulatory stuff is going to be the most important. That I'm confident in saying. Yeah, that I could definitely say.
Dr. Dan. Hi, it's nice to see you, and for first time I can meet you.
Yes,
Nice
hi
to meet you.
Dan.
Hey, Tangelo, how you doing?
Yeah, fine, thanks, and you?
I can't complain. Hanging out, talking about biology.
Yeah.
Uh,
I think
okay.
Leo left.
Any other issues that you have?
Oh, well, with the
Yeah.
eyeball thing, it's the same kind of thing. I mean, the human eye, right, has a lot of parts that, you know, our eyes don't work if you get rid of the muscles or the tendons or the lens or this or that. But an eye that's missing one or more of those parts is still perfectly useful, right? An eye that can't move but is otherwise the same. We can still tell the direction where light is coming from. That's useful. We can still form an image. We can, you know, move our heads around. I mean, bats, for example, can't move their eyes in their heads. So that's why their heads swivel like 180 degrees, right? Because they can't move their eyes relative to their heads, but they can move their head around. Like they can turn their head like they could do like an exorcist kind of move, right? They still work fine. So they're absolutely reducible. I mean, the big thing is this is just an irreducible complexity argument, and we've watched those types of systems evolve. We don't need to wonder if they can.
I think you asked me the question then, and you said that an irreducible system can evolve. But that is a contradiction in terms, because an irreducible complex system cannot evolve by definition. That is precisely the basis of the argument which Behe is making.
Then such systems don't exist, because we've observed the evolution of systems that meet his definition.
Well, give me an example. I can ask you, how did rhodopsin evolve?
How did what?
Rhodopsin evolve.
Oh, that's an easy one. Give me a hard one. Rhodopsin is all the opsins are a variant of a class of proteins called serpentine proteins that are signal receptors in the plasma membrane. It's a class of proteins called G protein coupled receptors. And way back before anything actually had eyes, there was a mutation in one of the variants of those genes that became sensitive to light rather than to a chemical substrate. Like, that's an easy pathway. We know how that one happened.
Okay, so
rather than play whack-a-mole, though, why don't we just talk about the logic of this hypothesis, right? Because Behe's hypothesis is that a system that meets a certain set of criteria cannot evolve, right? Well-matched interlocking parts that are required for a specific function, where if you remove any one part of the system, it ceases to function, and therefore it could not have evolved except as an intact unit all at once. Is that a fair representation of Behe's argument?
Yeah, I think a very important point to make clear here is that we are talking about the basic function of a system. Okay.
Right.
So of course, if you remove a part of a car like a piston, then you do not have anymore the basic function of even the simple of a fourth T. Like,
you are committing one of the major fallacies of Behe's argument, which is assuming that the function of the system must be the same throughout its evolutionary history, and that is false.
It's also worth mentioning that if one of the pistons in a car stops firing, it might not run the same way, but it can still technically run. In fact, they're developing cars now that when they're on the highway at a steady speed, a number of the pistons will actually shut off so that the car saves on fuel.
Right. Like Dr. Dan, you would agree, for instance, that at least a bunch of changes in evolution are co-opting certain structures and giving them new functions, right?
Yeah, 100%. Yeah. And then you can look at the new system and say, oh, that couldn't have evolved, because how could you get that specific function through a stepwise pathway where you're gradually increasing that function, having selection at each step of the way? That's not how evolution works. Selection pressures change over time, right? So what works in one place in time doesn't work in another. Functions of specific genes can change over time. Genes can you could have a change of a function, or you can just get a new function on top of an old one. And all of those things they're excluded from the original way that B, he described irreducible complexity. He only accounted for a very narrow set of evolutionary processes. And when you include how evolution actually works, turns out you have a lot of pathways to evolve these so-called irreducible structures. So either the cases that irreducibly complex structures can evolve, and we've observed it happening, or two, irreducibly complex structures do not exist. One of those two things is true.
It's almost as if there are so many ways for things to evolve and change that I wouldn't say evolution is or maybe I guess evolution is inevitable, since you have certain, or you have all this cascade of effects and lots of different pathways you can go, essentially. Like even now we were just talking about the different kinds of mutations and how there's so many different types even there.
Yeah, it's I wouldn't go so far to say as you could evolve anything necessarily, because they're like physical and physiological constraints on things. But in terms of like classes of things, it's difficult to describe a structure that you could not evolve, right? Like, could you come up with a physiological system? Again, you could be like, well, you could have a spider that's 3 stories tall and has blah, 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 you know, right? You could come up with a thing that doesn't work because like the exoskeleton would have to be too thick and it couldn't move, right? But like in terms of like what type of protein-protein interactions are could not happen through evolution, haven't found the answer yet. Like that's, yeah. I mean, I'd love to hear what Angela thinks of this, because, like, I mean, if you want, I can provide examples of structures and systems that meet Behe's definition that we've observed evolving. So I would love to hear what you think of this problem for your idea.
Okay, I have posted at your YouTube channel, at one of your videos, an example of irreducible complexity of the pathway to make chlorophyll. I don't know if you
read that. I read that, but here's the thing. That is an unfalsifiable argument, because you can always point to a system and say, we don't have enough information for me to say that this could have evolved. And when you get more information for that particular system, then you just pick something else that we haven't studied enough to find the answer yet. What I'm asking you about is the examples that directly falsify Behe's model, because what he's saying is if you meet these criteria, a thing cannot evolve. And I'm saying we've observed things that meet those criteria evolving. Therefore, his model is false. What's the response to that problem? The answer is not, oh, we've got these other things we haven't explained. That's whack-a-mole. That's not a falsifiable argument. That's a
Sorry, that's just too good.
Well, I need to demonstrate to you only one system which cannot evolve, and I think photosynthesis is a fantastic example, because without photosynthesis, you do not have advanced life. Okay?
How do we know it can't evolve?
Demonstrate that? How do you demonstrate that it can't evolve?
Well, I explained it to you in the post that in order to have the caption of photons, you need chlorophyll, which is the first step in the photosynthesis pathway. In order to have chlorophyll, you need 17 proteins and enzymes which produce chlorophyll. And you have in that pathway 10 enzymes which are also used to make hemoglobin.
Make it 100. It doesn't matter.
Yeah, can I finish, please?
I mean, we've all heard this argument before, honestly, though. It's an irreducible complexity argument, and it does nothing to address the problem that we've observed these systems evolving.
Then I would like to finish my speech, because you talked about 5 minutes and nobody interrupted you, and I hated to be interrupted.
What he said made sense, though.
So, okay, guys. Good night, everyone.
It's Angela. Go ahead. Nobody's interrupting. Go on. The thing is, right, like my response to that would have been, why is it exactly chlorophyll that needs to be the chromophore?
I was about to ask.
That's the answer.
That's the answer.
There's not one way to do everything. There's lots of ways to do everything.
Right? I mean, the system that's responsible, like, for the first step of that is a very large,
and you know how you know there's lots of ways to do that thing? the early steps in that biosynthetic pathway are almost identical to the early steps in the pathway to make the heme groups for hemoglobin that capture the oxygen that move in your blood within your bloodstream. Those chemical groups are very, very similar in terms of their biosynthetic pathways. It's likely they share a common ancestry. It's not definite, but it's very possible.
Yeah, I mean, to me,
So,
like that's interesting because when you study like photosynthesis as a pathway, you study to some extent the molecule that's starting it off, and it's like, well, that's a very giant, large aromatic system that is responsible for conjugating this photon along and transferring it, but doesn't seem like it's necessarily anything special. It
Here's
could
the
be
picture I wanted to find.
Yeah, it could be something else. It doesn't seem like it would have to be this way, but hey,
I
could be wrong.
I did want to say something really quickly. Otangelo has been sort of chomping at the bit, I guess you could say, to have a debate with the great Aaron Ra, yet he cannot stand being interrupted or overtalked. He'd last maybe 5 or 10 minutes in a debate with Aaron Ra before leaving.
Yeah, the thing is, Gavin's question. I mean, Gavin is a very one-trick pony, but there are things you can find in evolution. Well, there are things in the world you could find that could potentially falsify evolution. But yeah, I mean,
long list.
Yeah, like we just haven't found any of those things yet, and that's kind of the beauty of it. It's not.
Hey, Immutable.
Yeah, what's up?
At 8:13, what is this comment? And on the side, that's where this blocking.
Oh, okay. My bad. I'm sorry.
All he has to
I've
do
been
is
trying
just
to
scroll up and
find
hit the
time. Been trying to find the time to say something.
I want to know why they don't, and maybe they do, but I want to know why they don't let you set a timer on when you highlight a comment that after like 30 seconds it goes away. It's on its own. Maybe they do, and I don't know it, but I don't think they do.
The question to settle all questions. I mean, evolution's a really interesting topic, but I mean, Otangelo will always play hot potato in terms of what things constitute intelligent design and which things don't. I mean, the thing is, like, there's a lot of proteins that we probably don't understand fully their function. But the thing is, that's not sufficient ground to say that evolution doesn't occur, right?
Right. It's the defense keeps falling back, right? You're still like, well, we're making a stand here. Oh, we figured that one out. Now we're making a stand here. Now we're making a stand here. And you just keep falling back and back as we learn new things. And this is why, like, I feel bad Otangelo left. It's just that he doesn't have to give that chlorophyll spiel every time. I've literally heard that 5 or 6 times. Plus, we've had that exchange in text a number of times over the last few years. Otangelo, if you're still here, I'd love to chat. It's just like you say the same thing
monologue.
every time.
Really quickly,
it's like repeating the same thing, and the answer it's not addressing the question, which is Behe's model, the way he originally stated it, is falsifiable. That incarnation of the model has been falsified. Now the new version is not falsifiable. Like, address that problem, please, rather than continuing to try to find examples. That doesn't solve the falsifiability problem.
The thing is, like, I don't know if you were around since the beginning of the conversation, but when Otangelo came in, I was asking him if he understood, like, any aspect of, like, enzyme kinematics or any aspect of enzymology at all. Because one of the most basic aspects of protein function and how enzymes work as a class of proteins is that whenever you have a series of enzymes acting in parallel or, just as a sequence, essentially what you have is you have a continuing chain of the protein receiving a substrate, reacting it with something, forming something new. But the issue really, and the beauty really comes into the fact that proteins produce product at a specific rate. And typically it's not possible always for subsequent proteins to take in the exact amount that's being produced by its precursor. And that results in regulation. because certain products that are produced by one protein go back into previous pathways to inhibit other proteins.
Yep.
And you find that in certain certain pathways, like gluconeogenesis, it occurs several times. And it's one of these things where it's almost inevitable for a metabolic pathway to exhibit that type of behavior.
Yep.
And you really do wonder. It's like, okay, Otangelo, you're saying proteins can't evolve, or this thing had to be irreducibly complex, but you don't even understand the basics of how proteins fold, the basics that, there are specific motifs, like 3-5 residue amino acids, that if you have a certain sequence of them, that will influence the folding conformation of a protein. It might cause it to adopt, to form a helix, or it might cause it to do something else. But it's just very specific things are well studied, and it makes you wonder, why are you trying to discuss this thing as if though nobody is familiar with it? I mean, certain things with protein biochemistry we've understood for over 20 years now, but you make it sound like it's completely new.
Quick, either Immutable or Dan, give me an example of a major prediction that evolutionary theory made that we later confirmed.
Well, I mean, one of mine is the existence of Archaeopteryx, right? I mean, Darwin laid out an example of undifferentiated, of how a very specific pattern in, I believe it was bird, in the wings of birds or digits of birds were unfused.
Right, it's the loss of digits in bird wings compared to reptile limbs. Yeah, I should say non-avian reptile limbs. Yeah.
So this is a prediction that the evolutionary theory made that was later confirmed.
That we should find a specimen that has characteristics X, Y, and Z that is intermediate between modern reptiles, like non-avian reptiles, and modern birds, and boom. Yeah.
All right, Gavin, there you go. Because part of the scientific method is your model makes novel testable predictions that are later confirmed. So when you state that the scientific method hasn't been applied to evolution as a model, you're wrong. You're also wrong when you say, well, that's why they call it a theory. You obviously don't know what a scientific theory is. A scientific theory is a well-substantiated, rigorously tested, and repeatedly confirmed explanation of some observed phenomenon. But you don't really know much about science at all.
Specifically, theories provide a framework to make subsequent predictions that are then shown to be accurate. Yes. Right? So the most famous example of this probably is with relativity, where Einstein made a number of specific predictions that at the time we literally didn't have the technology to test.
Black holes.
But then subsequently in the 20th century, as we improved our technology, we were able to specifically test things like light bending around the sun, for example, from stars opposite side of the sun from us. And for evolution, you've got a lot of that stuff. For example, one of my, there's the morphological ones that there's just an endless list of these things. I mean, you could talk about the bird transition. You could talk about the not reptiles technically, but I think there were synapsid to mammal transitions. There's a beautiful record of that transition that was we're going to see this, and this. And we saw exactly those things. You could see it with the terrestrial vertebrate to whale transition series is just beautiful, where you see the gradual loss of hind limbs, the development of flippers from walking limbs, and my personal favorite part of that, the gradual migration of the nostrils to the top of the head. You can also see that in these skulls that are time ordered from most terrestrial to whales. It's really nice, but I really like the molecular stuff personally. So my personal favorite prediction was looking at non-human apes and the human genome and going, well, that's weird. We're missing a chromosome pair. What happened there? And by comparing all the pairs of chromosomes, we said, okay, there has to be a fusion somewhere. And we were able to find the chromosome that is a fusion. You could take human chromosome 2 and chimp chromosome 2p and 2q, and you stick them together end to end, and the banding patterns line up precisely. And not only that, but you can find the fusion site where there's these little end caps called telomeres that only exist at the end of chromosomes. And then there's subtelomere regions that only exist directly adjacent to telomeres. And smack in the middle of human chromosome 2, we've got subtelomere, telomere, and then flip the other way, telomere, subtelomere. Right where those two chimp chromosomes line up, you've got a dead telomere right in the middle of human chromosome 2. That is, and like we said, we're going to find a fusion. There's going to be two chimp chromosomes that line up with one human chromosome. Boom, right there, and it lines up perfectly.
Gavin just repeated the same thing. He hasn't been either. He's been listening, and it just is not capable. There's not enough brain cells in there to capture the knowledge, or he's just not listening at all.
So I hope everyone who's listening benefited from that, because
I did.
I think that stuff is cool.
Because I've always wondered, like, how precisely we know about the chromosome 2 fusion. You're pretty much saying it's like literally two puzzle pieces going together. Like,
and creationists love to say no, that's wrong, because there's a gene that spans the fusion site, and that's wrong. There's a pseudogene adjacent to the fusion site. It's a member of a family. There's a bunch of other examples of it in the genome. Every other member of that pseudogene family is adjacent to telomeres. It's always at the ends of chromosome. The only example in the middle of a chromosome is in the middle of chromosome 2, right next to the head-to-head telomere fusion. And the reason that creationists will say, well, it's expressed across that gap, that's wrong. There's an alternate start codon on the far side, so you sometimes extend the front end of that pseudogene, and you get a little bit of transcription across the telomere gap there. Okay. That's easy. There's lots of spurious transcription that goes on. That's not like hard. So that's and then it's not just the fusion site though. You line up the two chromosomes end to end, and they match all the parts of it. So you've probably seen, like, when you show like chromosomes, they've got like a banding pattern, right? They've got denser and less dense regions. You line up the two chimp chromosomes, boom, boom, with the human chromosome, the banding patterns match from top to bottom. They're the same. It's not like even close to debatable. It's beautiful.
I want to mention one in addition to the transitional species. Not frequently I've seen another examples. What about the modern transitional species? And that's very interesting. So why you don't talk for the modern? This is a good example for transitional species.
Modern.
Right now, today, transitional species.
Sure.
To go back to the fossils. Let's go a few examples. For example, the hippopotamus. I wish for Christmas hippopotamus, right? So this is a transitional species. It is obviously terrestrial and have aquatic life. It is transitional. So here's the question which you have to think about, and when you give example to creationists, you can use it. When, for example, hippopotamus will become something like manatee. How was this? Manatee.
Manatees, yeah, right.
So when? Now it will happen always when the landscape is changing, when you have another challenge. Then, for example, imagine Africa starts to be more wet, and there's less and less land. Therefore, the hippopotamus will become manatee slowly and clearly. And that's a good example for transitional species in front of your eyes. So in addition to that, I can give you another example. For example, the mud skipper. The mud skipper is a wonderful, beautiful transitional species from water to land. You can see it exactly in front of your eyes. Of course, if became more dry the landscape, the mud skipper will become more adapted to the here we go. That's my guy. So, and this is a good example again. What it needs for mud skipper to become fully land animal, it just needs changing of the environment to have another challenge, another pressure, environmental pressure. If you want, you can take a moose as an example. Moose is half in the water. chewing algae, some kind of the grass, underwater grass, and so on. Again, the moose may become completely aquatic if the landscape start to challenge this. However, it's also another prediction, Leo feels. The prediction which Darwin actually created about the so-called fossil, it's stuck in my mind. So when the organism doesn't change at all.
The stasis.
Yeah, stasis, I think.
When you don't have changing in the environment, when you don't have another challenge for survival, changing of environment, of course, everything is adapted. Living fossils. Here we go. I'm talking for living. Another prediction is the existence of living fossils.
Yeah,
So,
an example, for instance,
something that goes in this direction or another direction. It's just about adaptation. If there is no changing,
I'm sorry, Top Dog's thing is just making me.
So my point is, when you debate with creationists and try to say about transitional species, you can describe in front of our eyes transitional species, which is very good examples about that.
yeah, I mean, for instance, creationists would argue that coelacanths, for instance, are organisms that just seem to never evolve. When in fact it appears, like morphologically at least, and I could be wrong on this, they just appear to have been in stasis for a very long time. For a long time.
So
I actually recently learned that. Yeah, I'm sorry, I thought I was muted. I recently learned that there's actually pretty significant differences between the fossil ones and the living ones. I think I might have this backwards, and somebody can correct me at some point. But I think it was the fossil ones were coastal, and the modern ones are like deep sea, I think.
Really?
A lot of their morphology is very similar, but there's a few kind of telltales that are different between them. So it shows that their niche actually did change over however many millions of years. But it's still clear from the rest of their morphology that they're the same lineage. Give you another. I think that was a really important point that was just made, because creationists will often point to, well, what about things that don't change? Why are there still alligators, right? Well, it's because they're really good at their environment and they're not under pressure to change their ecological niche. But when things become under pressure to change their ecological niche, that's when you see new traits, right? And it kind of goes back to this problem with Behe, where he's assuming that like same function, same selective pressures, whatever, where it's like, no, everything is dynamic all the time. So you're going to have a tug of war stay the same, get different. It's going to change over all these long periods of time. And I'll add one more example, and then we're going to go, because I don't want to hug the show, and I enjoy listening to all of you. But
I mean, we're about to go live again, actually, because we're doing an after show for Leah Phillips' debate he had with Joe on the Raging Atheist channel, I think.
Yeah.
Is that tonight?
Starts at 8. We had a discussion about dualism versus physicalism as it relates to the mind, and then idealism versus realism.
Nice. That sounds interesting. So let me just give you this one example before I go. There are lizards right now. There's at least, I think there are 2 different species that are not just evolving the ability to give live birth as opposed to laying eggs. There's a bunch of lizards that do that. That's evolved a bunch of times. But I think there's 2 examples right now that are actually evolving true placentas. And there's like actual invasive placentas. So instead of basically developing an egg without a shell, it's getting nutrients from the mother rather than the yolk via the uterine wall, which is a new thing in reptiles. No other reptiles, have true placentas. This is a thing that's happening right now. And in one of the populations, there was an example where a lizard laid 2, you want to talk about a transition? Check this out. A lizard laid 2 eggs and then gave live birth to a 3rd offspring in the same litter.
Wow.
Huh.
Yeah.
Science.
So how's that for an evolutionary transition, right? It's like yeah. So that's like an ongoing thing right now, where you've got lizards that are that now have placentas, which is obviously a defining characteristic of mammals, right? So that's going on like right now. It's not hypothetical. It's not on the fossil record. We're watching this happen right now.
Well, and I think we're actually going to stop, but I do want to kind of compliment that by saying, I guess, and this is true for even people that are kind of just getting their toes into evolution, but it's that sometimes we're tempted to think that an organism and the landscape or ecosystem which it inhabits are two different things. But in fact, like when we talk about evolution in organisms and changing over time, there really is, at least in the course of like thousands or millions of years, like an additional context where we need to talk about how those organisms change within the context of their environments as well. Because, like you mentioned, like there's certain organisms, like alligators, for instance, that may just never change because they're too stable for their environment, and their environment just may not change in such a dramatic way that would, really necessitate that much of a, genetic makeover. But there are organisms, for instance, that when the environment does change, they do have to change their reproductive strategies, or certain things do have to change in terms of metabolism or whatever in order to accommodate that new environment. And over time, those changes may be heritable and cause speciation within, whatever organism we're discussing.
Before you go, Dan, ARE did want to ask you a question about phylogenetics.
Say that again?
A rational empiricist wanted to ask you a question about phylogenetics.
Oh, cool.
Sorry, I always have a lot of questions for you, Dan, just because.
Please go for it.
So I guess my last question would be, you were talking about transitions, and it kind of got me thinking about phylogenetic trees and how they're constructed. And I was just wondering if molecular or trees based on molecular data today, are any of them constructed using any phonetic methods, or are most of it based on cladistic methods like likelihood analysis and such?
It's, to my knowledge, I only have experience with cladistic methods. So, and there's lots of variants of that. It depends on how you construct your model, right? There's maximum likelihood, there's parsimony, but that's like kind of no one really does that. That's like what you learn in class, and then no one does that for real. A lot of the techniques now are using like Bayesian kind of modeling. So you're doing like iterative. You have a set of priors, a set of parameters that you're saying in terms of like base specific mutation rates and population size and that kind of stuff. And then given that set of parameters, some of which you can observe and some of which might be inferred depending on the situation. And then you can say, okay, here are the parameters. Here's all your, alignments. Go, model, build me a tree that matches these parameters best. And so you'll generate like 10,000 trees, and then pick like the top however many that are good, and then perturb those and generate 10,000 variants based on those, and then select the best ones from those in terms of how well they match, and do that iteratively like 1,000 or 10,000 times until you get a tree that fits your parameters very well. And then the result of that is going to be a nested hierarchy where you have a bunch of extant things, right? A bunch of living things, and then you're grouping them based on hypothetical ancestral sequences that are the consensus based on the parameters of your model in terms of like mutation rates and that kind of stuff.
on synapomorphies?
Yeah, well, yes, but it's molecular. It's important that it's molecular synapomorphy. So it's like specific nucleotides at specific sites is what you care about.
And those methods can also be applied to fossil evidence as well, correct? The
yes,
likelihood.
Absolutely, but that's trickier, right? Because it's really easy for like a DNA sequence, like, okay, we're gonna decide that A is the ancestral nucleotide at a site, and then like T is the derived nucleotide at a site. Like that's computationally very simple to deal with, and then to a lesser extent, like insertions and deletions you can deal with. But like for morphological traits, it's trickier, right? It's like, well, femur length, like, how do you what's ancestral and what's derived, and where do you draw that line? It's a trickier thing to quantify when you're using morphology. You also have to decide which traits morphologically, and sometimes they're in conflict with each other, which makes it fun.
Okay, thank you.
Also,
You're welcome. I hope that at least part answered your question.
So, in addition, there is also some tricky moment when you do phylogenetic tree, for example, for very tricky proteins like amino acid synthetases, for example. Those are very difficult to trace by sequence. If you go in a sequence, you almost you'll find for sure motifs, which is equal, and you can align those motifs. But still, it's not sufficient and enough to make all the amino acid synthetases just to make them in a good phylogenetic tree. However, there is another program which is called Philo2, maybe it's called, which is more structural base.
Mm-hmm.
So you're using a structural base phylogenetic, and also you can build up a very cool, most probable, let's say this way, most probable phylogenetic tree for amino acid synthetases. So for few proteins, there is a tricky moment. So you don't always go based on the sequence. The same is true for the ribosome, for example. Ribosome also is very tricky in the sequence. Yes, there is some core of the sequence which is almost identical in all of the domains of life. However, if you go for the branches and different loops around in a ribosome, you might not see a very good sequence alignment. So again, you have to base on some kind of structural based system.
Yeah, I guess I was only asking because when I asked the same question or a similar one that I just asked you, Dan, to a different biologist, he basically said something along the lines of DNA characters, the nucleotides, don't show ancestor descendant polarity or something like that, and that like because they're usually compared against like extant species, and so I was kind of confused by what he meant by that.
That's true, because we don't have like fossil DNA, right? We can't, we can tell how, say, for example, humans and cartilaginous fish, right, are related based on the similarity of their DNA sequences compared to the similarity of like humans and reptiles or humans and amphibians or whatever you want to do. But we don't have that ancestral DNA sequence to compare, which is fine because we have the techniques to be able to figure out how to infer that, because we can figure out, for example, like how fast does A mutate into G? How fast does it mutate into T? How fast does it mutate into C? How fast does G mutate back into A? We have these types of things. We can like count those. We could figure that out. And then you could say, okay, applying that rate, those parameters to all the differences we observe, what's the most likely pathway working backwards to a common ancestor sequence? And that doesn't necessarily mean the fewest mutations. It means what's the most likely pathway that common ancestor took to get to the sequences that we have today based on these parameters that we can quantify. So yeah, it's not that we're saying the idea is that nothing alive today is ancestral in terms of like its lineage to anything else that is alive today. But by doing the comparisons, you can work backwards and figure out what those common ancestors would have looked like genetically.
Okay.
That's the idea.
Thank you.
I hope that helped. So, go ahead, Speed. Sorry, I didn't mean to cut you off.
Something that I'm very interested in is in say a complex organism, in finding alternative pathways, basically biochemical pathways, and in finding biochemical dead ends. Because I think, according to what my hypothesis, that there are all sorts of little pathways that are neutral right now, and they don't go anywhere, but at some point they can lock into something. So that would be go to the evolution of biochemical pathways. And then what you were talking about is these phylogenetic pathways. where and those are usually done with gene regulation. Will make a change in the how the embryo develops, and you'll find basically a pathway to the phenotype. Okay, phenotypes that there's a phenotypic pathway from one species to another, and discovering those is going to be kind of fruitful. But I think those biochemical pathways would be quite fascinating. I think there are a lot of them that don't go anywhere. They're dead ends or they're alternatives already to some existing biochemical pathway.
I think to some extent, so I think some biochemical
I'm gonna head
pathways.
out. Bye, everybody. Thank you for letting
Thank you,
me
Dr.
hang out
Dan.
and chat with you.
Dr. Dan,
Always fun.
Yep.
See ya.
what happened to Otangelo?
Oh, he left. He got triggered.
Triggered?
Yep.
Okay, so we're supposed to be on another stream now.
Well, I'm going to answer your question, then we're going to go off live. Going to go off air.
Okay.
I think to answer your own question, and Demeter knows more about this than I do, so he probably could have a better opinion. I think a lot of biochemical pathways have evolved in order to, and not just for metabolism. Metabolism obviously is important, but to kind of deal with regulation and deal with cell signaling in order to kind of help things in terms of managing complexity in a way. So I'm not sure if it's dead ends necessarily, as much as it is trying to make sure that the whole system is kept intact.
Yeah. Well, there may be some developing biochemical pathways at dead end. They don't actually do anything functional right now, but they're like one step away from fitting into another, locking into another pathway. And these full cell assays of proteins and mRNA or small RNAs will yield a great deal of information about all that. But what I hypothesize is that there are a lot of these pathways. We're going to find them in small amounts, and they haven't quite locked in yet. And I think that's how evolution of complexity happened, that there are a lot of these pathways out there, but it would be interesting. So
don't forget that many of the products, so you have substrate and products in, for example, classical biochemistry, many of those products became very similar. And in fact, the way to jump, let me say, one methylation to some product. So you have products and substrate and product. So, and in order the product to become substrate for another enzyme, it could be just a very small step in terms of, let's say, one methylation or acetylation or something like that. But in fact, similar domains that do that are frequent. They're everywhere. So you just need the right domain to transfer by mutation, to transfer the particular domain in the particular section, and voila, you have new functional enzyme.
It depends on what kind of a network it is.
Yeah. So you are correct, but you're assuming too far away. Maybe they're not. So in fact, they're not too far away between each other. Of course, it all depends on the selection after all. So is this product will be beneficial somehow, and in the right conditions might be beneficial.
Yeah.
So
or the development of a parallel biochemical pathway, and then the original pathway isn't necessary anymore.
And also there are lots of, for example, signaling molecules which goes through the post-translational modifications.
Yeah.
Like glycosylation or different types of modifications, or even just digestion by proteases.
Yep.
So, and that's very frequent as well.
So you guys all ready to hop off this one and hop on another one?
Yeah.
Okay,
I was ready 8 minutes ago.
Yeah, so I will leaving, and I might listen the second part, but I might not be involved. So
Okay.
All right. Well, we will get going. Thank you all for joining. Thank you all for listening. And we will see you later.
Yep. Thank you. Thank you all. Okay. I'm going to end, or are you ending? I'm ending.